There are several separate populations of eyeless A. mexicanus in different caves. There are populations that spend time in areas with light, and the fish in these populations have eyes. So that means we've got a population of the same species without eyes and a population of the same species with eyes, depending on where they're located, whether light is present. The mitochondrial DNA, mtDNA, of the eyeless A. mexicanus was compared with the mtDNA of the A. mexicanus with eyes. So this is another way to compare the genetic relatedness between two organisms using DNA hybridization. So they tried to hybridize the mitochondrial DNA from the two species or the two organisms. In this case, the two organisms of the same species. Suggest how DNA evidence can help to find out whether the eyeless A. mexicanus and the A. mexicanus with the eyes are the same species or they are the different species. So you know that you can use DNA hybridization to find out the genetic relatedness between the two organisms here. Okay. So how, the question is how the evidence from the DNA hybridization will give you the information so for you to conclude whether they're the same species or different species. Keyword of the question is how. So how do you go about doing the conclusion from your DNA hybridization? So let's review your theory first. Okay, using this opportunity to review the theory for DNA hybridization. So in DNA hybridization, we obtain the DNA from the two organisms that we want to compare. Okay, in this case, it's species A and species B DNA, but in the question, it's the same species growing in different area, one with light and without light. So we obtain their DNA, and then we heat the DNA in order to break the hydrogen bonds. So all the DNA becomes single-stranded. And then we mix the DNA from these two organisms together so that they will form complementary base pairing. So the more bases that are complementary, the more hydrogen bonds will be formed between them. And the next step is we do a temperature denaturation. That means we subject this DNA to heating, to high temperature. So slowly increase the temperature to see what is the temperature that's needed to denature this DNA. So the higher is the temperature, that means the more hydrogen bonds you need to break. So that means the higher is the temperature, there are more bases that can form complementary base pairing. So that means there are more similarity between the two DNA. So the temperature itself will be able to tell you the number of nucleotides that are similar between each other. Okay. So for the question so how do we obtain the evidence to make the conclusion is we count the number of bases or the nucleotides. This is a very important word that you need to have in your answer to score. Based on the temperature denaturation, we can find out and count the number of bases where base sequences that are different. So we'll know how many base sequences that are different. Okay. So the lower the temperature, that means the more the differences in the base sequence. So compared to original DNA, what is the temperature that's needed? And as the temperature needed to denature decreases, there'll be more and more bases that the sequences are not similar. Okay. So when you count them, when you got fewer differences, in the empty DNA sequences. So we are looking at the base sequences or DNA sequences. We have to say the fewer differences in their sequence or the nucleotides or the bases suggest that they are the same species. Okay. So that's the answer. So same species or different species. First, you must count the differences in the base sequence. Count the number of bases that are different, and the less differences suggest that they are the same species. Okay. Right. Next question. Suggests why empty DNA is used instead of nuclear DNA when studying the closeness of the relationship between two populations. So this is a very important question, 3 marks. So going back to the theory, here. So over here in this PowerPoint, I have actually explained to you why is it that we need to use the advantage of using mitochondrial DNA versus the DNA from the nucleus of the cell. First thing is because the human mitochondrial DNA only comes from the mother, right? Only mother, the whole cell is present with the mitochondrion that's passed from one generation to the next. So therefore, when it comes from a female line, there will be less genetic variation with subsequent generations. So that will allow us to be able to see genetic relatedness because there's less changes in the genetic makeup of the mitochondrial DNA that's passed on from mother to the offsprings. Okay. And then the empty DNA is circular. So it's a circular DNA, mitochondrial DNA. So when it's circular DNA is like the bacterial DNA. There is no crossing over during cell division. So it will not lead to changes in genetic makeup. So the genetic makeup of this DNA is quite stable throughout generations. So this is what we want to achieve in point 1 and point 2. Stability of the genetic makeup is maintained with generations. All right. So the only thing that will lead to the changes in the genetic makeup is, of course, the mutation. Okay. It is said that the mutation in the mitochondrial DNA is quite constant. And it mutates faster than the nuclear DNA. That means the rate of mutation is 1 in 25,000 years for nuclear DNA. So it's very, very slow, right? Whereas for mitochondrial DNA is faster because this circular DNA from bacteria, they have no histone protein around them. So they're not protected from histone protein. And then in mitochondrion is an organelle that carry out aerobic respiration. So oxidative phosphorylation stage of aerobic respiration involves oxygen. So a lot of oxygens will be found in mitochondrion, which is a mutagen, which will oxidize this mitochondrial DNA. So therefore, mitochondrial DNA will change due to mutation. So the rate of change of this mitochondrial DNA is constant. So there are certain rate that it changes. So with that changes, we can actually detect how different there is between these two organisms because of mutation. Okay, so what is the answer that's needed for you to write in order to score for this question? Is why do we choose empty DNA? Because the empty DNA is inherited from mother only. Okay, and there is no crossing over to produce genetic variation or to change the genetic makeup of the mitochondrial DNA. Okay. then the mitochondrial DNA mutate faster than nuclear DNA. So you can see the changes in a short period of time when you want to compare between the two organisms. Okay, and the mutation of the mitochondrial DNA. Okay, what happened? The mutation in mitochondrial DNA is, I write again, mutation in mitochondrial DNA is constant. So you can base on the rate of mutation to know how long they are subjected to the natural selection process from the original ancestral species. And of course, there are many copies of mitochondrial DNA in the mitochondria, many copies of mitochondrial DNA per cell. Because one cell can have many mitochondria. So when you want to isolate the DNA, you will be able to get many copies of them for your analysis. So this is a very important point. Not associated with histone protein. Okay, so that will reduce, therefore increase chance of mutation. Okay. So basically, these are the answers that you can write for why we use mitochondrial DNA. Right, the numbers are huge, so easy to isolate, so can obtain the results faster is also a marking point. mtDNA analysis is faster because all the mitochondrial DNA are the same, right? So therefore, and there are many copies in the cell, so faster to isolate and very easy to check the differences between them. Compared to if you get the DNA from the nucleus, all the DNA are different. We have got, let's say, 46 chromosomes. For every chromosomes, the base sequences can be different. Okay, so therefore, there are too much differences for us to compare. So it makes the analysis becomes more difficult and longer. Right, so that's all for the discussion for today.